Hello friends and welcome. Today I am going to deliver lecture on T cell maturation in thymus, thymic selection and cytokines properties and functions. Let us first try to have a look at the main objectives of today's deliberation. To have a detailed idea about maturation of T cells in thymus and the process of thymic selection, to know about SelfMHC restriction of T cells and what T cell receptor complex is, to have general idea about cytokines, to discuss properties and functions of interferons and interleukins, that is IL-1, IL-2, IL-4. Now we will start with the brief idea about T cell maturation. T cell maturation occurs in thymus. Progenitor T cell from the early sites of hematopoiesis begins to migrate to the thymus at about day 11 of gestation in mice and in the 8th or 9th week of gestation in humans, in a manner similar to B cell maturation in the bone marrow. T cell maturation involves rearrangements of the germline TCR genes and the expression of various membrane markers. In the thymus, developing T cells known as thymocytes proliferate and differentiate along developmental pathways that generate functionally distinct subpopulation of mature T cells. The thymus occupies a central role in T cell biology. Aside from being the main source of all T cells, it is where T cells diversify and then are shaped into an effective primary T cell repertoire by an extraordinary pair of selection processes. The most immature thymocyte expresses neither core receptor and is referred to as CD4 negative, CD8 negative or double negative, that is DN cells. These progress to the CD4 positive, CD8 positive, that is double positive or DP stage, which in turn mature to the CD4 positive, CD8 negative or CD4 negative, CD8 positive, single positive or SP stages. Now let us try to understand thymic selection. The recognition of what is self, that is what belongs to one's own body, and what are non-self or foreign, is the fundamental property of the immune system. The achieving of this self-tolerance occurs during the T cell maturation in the thymus. Surprisingly, nearly 95% of all the cells form their dye before they are ready to mature and immigrate into the peripheral lymphatic organs. This wastefulness is due to the extremely stringent selection criteria termed thymic selection that are applied to the T cells that develop in the thymus. This way, it is made sure that the only T cells with potentially useful receptors survive. Although a few self-reacting B cells are also eliminated during their development, the main mechanism of self-tolerance proper lies, nevertheless, in the elimination of developing self-reacting T cells in the thymus. Double positive pre-T cells undergo two selection processes in the thymus, positive and negative selection. In case of positive selection, the double positive pre-T cell interacts with SelfMHC molecules expressed on epithelial cells in the thymic cortex. This interaction results in the survival and differentiation of the double positive cells. Double positive cells that do not make this critical interaction and are thus not selected die by apoptosis. Thus, in positive selection, double positive T cell becomes educated to the MHC molecules expressed by the thymic cortical epithelial cells. This means that for the rest of the life of the T cell, even as a mature cell, when it leaves the thymus, it will respond to antigen only when the antigen is bound to the MHC molecules that the developing T cell encountered in the thymus. Whereas negative selection of T cell works by inducing apoptotic death of cells whose receptors recognize self-peptide antigen MHC complex in the thymus with high affinity. Current evidence indicates that peptides expressed by the cortical epithelial cells play a critical role in the positive selection setup. These peptides are derived from self-antigens either expressed in the thymus or brought in the thymus. It is not currently clear, however, how these peptides derived from self-antigens select T cells with TCR specificities for non-self as well as self-antigens. Now let us discuss the next part of our today's lecture, that is SelfMHC restriction of T cells. MHC restricted antigen recognition or MHC restriction refers to the fact that a given T cell can interact with both the self-major histocompatibility complex molecule and the foreign peptide that is bound to it, but will recognize and respond to the antigen only when it is bound to a particular MHC molecule. When foreign proteins enter a cell, they are broken into smaller pieces called peptides. These peptides, also known as antigens, can derive from pathogens such as viruses or intracellular bacteria. Foreign peptides are brought to the surface of the cell and presented to cells by proteins called the major histocompatibility complex, that is MHC. During T cell development, T cells go through a selection process in the thymus to ensure that the T cell receptor, TCR, will not recognize MHC molecule presenting self-antigens. The selection process results in developed T cells with specific TCRs that might not only respond to certain MHC molecules but not others. The fact that the TCR will recognize only some MHC molecules but not others contributes to MHC restriction. Both CD4 positive and CD8 positive T cells can recognize antigen only when it is presented by a self-MHC molecule, an attribute called self-MHC restriction. Beginning in mid-1970s, experiments conducted by a number of researchers demonstrated self-MHC restriction in T cell recognition. A. Rosenthal and E. Schwarz, for example, showed that antigen-specific proliferation of Th cells occurred only in response to antigen presented by macrophages of the same MHC haplotype as the T cells. Now let us try to learn about T cell receptor complex. T cells, like B cells, express antigen-specific receptors. The T cell receptor, that is TCR, is a heterodimer and composed of two transmembrane glycoprotein chains, alpha and beta. The extracellular portion of each chain consists of two domains, resembling immunoglobulin variable, that is V, and constant C domains respectively. Both chains are glycosylated and connected with each other with the help of interchain disulfide bond. The transmembrane helices of both chains are unusual in containing positively charged, that is basic, amino acid residues with the hydrophobic transmembrane segment. The alpha chain carry two such residues, the beta chains have one. The antigen recognizing alpha and beta chains of the TCR are also expressed on the surface of T cells in tight non-covalent association with CD3 molecule. The combination of the alpha and the beta chains of the TCR and CD3 is referred to as the T cell receptor complex, analogous to the B cell receptor complex. CD3 is a complex of 5 invariant polypeptide chains that associates to form 3 dimers, a heterodimer of gamma and epsilon chains, a heterodimer of delta and epsilon chains, and a homodimer of 2 zeta chains, or a heterodimer of zeta and eta chains. CD3 acts as a chaperone and helps in transporting the newly synthesized TCR molecule through the cell-to-cell surface. Let us try to learn about cytokines. First of all, let us have a general idea about cytokines. Cytokines are low molecular weight, generally less than 30 kilodalton, soluble proteins, glycoproteins, non-immunoglobulin in nature, produced and secreted by a variety of cell types and act non-enzymatically through specific receptors to regulate host cell functions. They play major roles in the development of cellular and humoral immune responses, induction of inflammatory response, regulation of hematopoiesis, control of cellular proliferation and differentiation. Cytokines can affect the same cell responsible for their production, that is an autocrine function, or nearby cells, a parakine function, or they can be distributed by the circulatory system of the distinct target cells, an endocrine function. Cytokines produce biological actions only when they bind to specific high-affinity receptors on the surface of target cells. Interferons and interleukins are various types of cytokines. Moving on to interferons. Interferons are a group of signaling proteins made and released by host cells in response to the presence of several pathogens such as viruses, bacteria, parasites, and also tumor cells. In a typical scenario, a virus-infected cell will release interferons causing nearby cells to heighten their antiviral defenses. Interferons belong to a large class of proteins known as cytokines, molecules used for communication between cells to trigger the protective defenses of the immune system that help eradicate pathogens. More than 20 distinct interferon genes and proteins have been identified in animals, including humans. They are typically divided among three classes: type 1 interferon, type 2 interferon, and type 3 interferon. Interferons belong to all the three classes, are important for fighting viral infections and for the regulation of the immune system. Let us first of all discuss properties of interferons. The basic properties of interferons are: number 1. Interferon are heterogeneous class of proteins with a molecular weight of about 63 kilodalton. Its molecules contain following components: tyrosine 2.3%, tryptophan 2.6%, arginine 7.2%, lysine 11.1%. It does not contain nucleic acids and only traces of carbohydrates. Number 2. It does not appear to be antigenic, and in the tests so far carried out with both concentrated and crude specimens has shown no signs of toxicity. Number 3. It shows tissue and species specificity. Number 4. Insensitivity to the action of virus neutralizing antibodies. Number 5. Resistance to the action of an acid medium and thermostability at 56 degrees Celsius. Number 6. Sensitive to proteolytic enzymes. Number 7. Ability to inhibit reproduction of certain viruses in tissue cultures, that is, absence of virus specificity. Number 8. Molecular weight of interferons depends both on the properties of inducing agents and on the cell producing the inhibitor. However, it has so far proved impossible to disintegrate the heavy interferon into the light. It apparently does not interfere with the body's natural immunity mechanism. Broadly speaking, interferon is produced when many different types of living cells, normal or malignant, are incubated with live or inactivated viruses. It can be produced in kidney tissue, testes, amnion, lung, and chorioallantoic membrane from human and monkey tissues, as well as from chick, rabbit, and many other animal species. Moving on to functions of interferons. All interferons share several common functions. They are antiviral agents and can fight tumors. As an infected cell dies from a cytolytic virus, viral particles are released that can infect nearby cells. Number 1. Interferons induce production of hundreds of other proteins known collectively as interferon-stimulated genes, ISGs, that have roles in combating viruses. Number 2. They also limit viral spread by increasing p53 activity, which kills virus-infected cells by promoting apoptosis. Number 3. Another function of interferons is to upregulate major histocompatibility complex molecules MHC first and MHC second and increase immune proteasome activity. Number 4. Interferons such as interferon gamma directly activate other immune cells such as macrophages and natural killer cells. Number 5. By interacting with their specific receptors, interferons activate signal transducer and activator of transcription, that is STAT complexes. STATs are a family of transcription factors that regulate the expression of certain immune system genes. In addition to the JAK-STAT pathway, interferons can activate several other signaling cascades. Both type 1 and type 2 interferons activate a member of CRK family of adapter proteins called CRKL, a nuclear adapter for STAT5 that also regulates signaling through the C3G RAP1 pathway. Interferons increase expression of class 1 MHC molecules and thus promote recognition by cytotoxic T cells. They can activate natural killer cells and can kill virus-infected cells. They play an important role in the first line of defense against viral infections. Interferon alpha induces an antiviral state in most nucleated cells. Since interferons have anti-proliferative effects that have also been used to treat certain tumors such as melanoma and Kaposi's sarcoma. Now let us move to interleukins. Interleukins are a group of naturally occurring proteins that mediate communication between cells. Interleukins regulate cell growth, differentiation, and motility. They are particularly important in stimulating immune responses such as inflammation. Interleukins are a subset of a large group of cellular messenger molecules called cytokines, which are modulators of cellular behavior. Like other cytokines, interleukins are not stored within cells, but are in a state secreted rapidly and briefly in response to a stimulus such as an infectious agent. Once an interleukin has been produced, it travels to its target cell and binds to it through a receptor molecule on the cell's surface. This interaction triggers a cascade of signals within the target cell that ultimately alter the cell's behavior. The first interleukin were identified in the 1970s. Initially, investigators believed that interleukins were made chiefly by leukocytes, white blood cells, to act primarily on other leukocytes, and for this reason they named them interleukins, meaning between leukocytes. Now let us discuss last section of today's topic, that is properties and functions of interleukins, which include interleukin 1, interleukin 2 and interleukin 4. Interleukin 1, that is IL-1, known previously as lymphocyte activating factor, that is LAF, is primarily an inflammatory cytokine. Biologically, interleukin 1 is more closely related to tumor necrosis factor TNF than any other interleukin, although the structure and receptors for interleukin 1 and tumor necrosis factor are clearly distinct. Interleukin 1 is known to be an important mediator of the immune system, produced primarily by mononuclear phagocytes in response to injury and infection. Interleukin 1 is also produced by non-phagocytic cells such as keratinocytes, Langerhans cells, corneal epithelial cells, endothelial cells, and astrocytes. Interleukin 1 alpha and interleukin 1 beta are two forms of the same class of interleukin 1. Although the amino acid homology between interleukin 1 alpha and interleukin 1 beta is limited, 26% homology. Both interact with the same receptor and appear to have identical biological activities. A closely related protein, ILI-RA, acts as an antagonist by binding to ILI's receptors. These two forms of ILI are initially synthesized as 31 kilodalton precursors and are cleaved by proteases to release mature, biologically active 17 kilodalton proteins. They are structurally similar and appear to carry out the same function by binding to a common receptor. One of the most important activities of interleukin 1 is activation of T lymphocytes during antigen presentation. Antigen-specific activation of T cells depend on two distinct but interrelated macrophage signals. The first signal requires the macrophage to internalize, process and present the antigen on its cell membrane bound to MHC. The second signal depends on macrophage production of interleukin 1 and interleukin 6. Interleukin 1 has two effects on T lymphocytes. A. Interleukin 1 stimulates synthesis and secretion of interleukin 2 by T cells. And B. Interleukin 1 also induces synthesis of interleukin 2 receptors, which in turn allow the T cell to respond to interleukin 2 by undergoing clonal expansion. In addition to interleukin 2, activated T lymphocytes produce interleukin 3, that is IL-3, multi-colony stimulating factor. Interleukin 4, IL-4, B cell stimulating factor first. Interleukin 5, that is IL-5, B cell growth factor, and interleukin 6, that is IL-6, B cell stimulating factor. Interleukin 1, also known as endogenous pyrogen, is related functionally to TNF and interleukin 6 in that all three are important mediators of both local and general inflammatory responses. Now let us discuss interleukin 2. Interleukin 2, first described as T cell growth factor, is a 15 kilodalton glycoprotein. The protein is secreted as a single glycosylated polypeptide, and cleavage of a single sequence is required for its activity. NMR studies suggest that The structure of interleukin 2 comprises a bundle of 4 helices, termed A to D, flanked by 2 shorter helices and several poorly defined loops. Residues in helix A and in the loop region between helices A and B are important for receptor binding. Secondary structure analysis has suggested similarity to interleukin 4 and granulocyte macrophage colony stimulating factor GM-CSF. One of the most important functions of interleukin 2 is its ability to stimulate clonal expansion of activated T lymphocytes. Receptors for interleukin 2 are present in extremely low concentrations on resting T cell. Now let us discuss interleukin 4. Interleukin 4 is highly pleiotropic cytokine. It is a 15 to 20 kilodalton 129 amino acid glycoprotein secreted by activated T lymphocytes, mast cells and basophils, and bone marrow stromal cells. Human and murine interleukin 4 share sequence homology of 41%. The biological and receptor binding activity is species specific. Recombinant interleukin 4 contains 6 cysteine residues which form disulfide bond. The interleukin 4 has growth inhibitor effects on many solid tumor cells types in humans. Interleukin 4 has a compact globular folded structure similar to other cytokines, stabilized by 3 disulfide bonds. One half of the structure is dominated by a 4-alpha helix bundle with a left-handed twist. The helices are antiparallel with 2 overhand connections, which fall into a 2-stranded antiparallel beta sheet. The receptor for interleukin 4 is known as the interleukin 4 R-alpha. This receptor exists in 3 different complexes throughout the body. Interleukin 4 has recently been shown to stimulate fibroblast proliferation and may mediate fibrosis in chronic inflammatory disease. Interleukin 4 appears to play an important role in the downregulation of interleukin 2 induced natural killer cell and LAK cell activities. Incubation of natural killer cells with R interleukin 2 results in activation and proliferation, leading to increased tumor cytotoxicity. When natural killer cells are cultured with both interleukin 2 and interleukin 4, there is marked reduction of interleukin 2 enhanced cytotoxic activity. That is all about today's lecture. Hope you have understood it well. Thank you very much and goodbye.